Hello and welcome back to another online lecture. So today we have graduated from topic 15 and we are going to start with topic 16. And topic 16 is all about anaerobic respiration and fermentation. And I'm going to go ahead and apologize in case you hear any noise in the background. I swear everyone in my neighborhood waits to cut their grass or to weed whack or to make loud noises outside when I'm filming. So if you hear any of that, I want to just go ahead and apologize. So I hope you all are having a great day regardless of what day it is that you are watching this. Currently it is Saturday, March 28th and it's early afternoon. So what we are going to talk about in this topic is we're going to talk about some basic definitions and we're going to answer the question of why do we even care about anaerobic respiration and fermentation? Why is it so important? We will walk through anaerobic respiration and then we're going to talk about two types of fermentation. And for this topic, our overarching question that we're working on answering is how do we get energy from glucose if no oxygen is available? So let's go ahead and start with some definitions. So if you recall from aerobic respiration, when we talk about cell respiration deals with a metabolic process that converts nutrients to energy. So we should already be familiar with what aerobic respiration is, and this is a form of cellular respiration that requires oxygen, so that arrow tells us oxygen. Then we have anaero, which means no. So anaerobic respiration is a form of cellular respiration which does not use oxygen. However, as we will see, even though anaerobic respiration does not use oxygen, we still use the electron transport chain. So we'll do a bit of a compare and contrast between aerobic and anaerobic respiration. And then finally, for fermentation is an anaerobic pathway that does not involve the electron transport chain. Okay, so these are a lot of definitions, and I'm going to go ahead and on a new page here, we're going to draw a little flowchart. So you all know that I like to try to put things pictorially or into charts that help us keep things organized. So when we have cellular respiration, we can break cellular respiration into kind of two pathways. We can have aerobic pathways, and we can have anaerobic pathways. So for our aerobic pathways, the aerobic pathway that we have talked about is aerobic respiration. So this was topic 15. On the right, for anaerobic pathways, we can actually split that into two, and we can split that into anaerobic respiration and fermentation, both of which will be covered in this topic. So again, this is just kind of putting everything that we just said on the previous page into a pictorial version. So let's go ahead and first talk about why are anaerobic respiration and fermentation so important? So I have a bunch of pictures here, and as we actually go through the different types of fermentation and anaerobic respiration, I'll actually give you more examples as well. So first and foremost, oxygen isn't everywhere. So if you remember back to Bio161, when we talk about all of the different waters and we talk about the different layers that are found in lakes and in oceans, the further down you go, the less oxygen there is. But there's still life there. And so that means that there are organisms that have to have a way of doing cellular respiration that is not aerobic respiration. And so another thing we can think about is this idea of our bodies. So when we are working out strenuously, we don't always have enough oxygen. And so how are our muscles still obtaining energy? And we'll actually answer that question. First, lots of drinks. So here we've got wine. Here we have a whole bunch of different Polish beers. So as you all know, I'm Polish. So I like to throw in my Polish background when I can. These are probably the most popular Polish beers. Żywiec right here, and Okocim is not on here, but Żywiec and Okocim, which usually has an OK, so a lot of my husband and a lot of our friends here like to call it OK beer, but those are probably the two most popular ones in Poland. And you can occasionally actually find them here in America as well. So we use anaerobic respiration, fermentation. These processes are used to make food and drink. So here's sauerkraut. My family actually also makes sauerkraut. So here's a jar of our homemade sauerkraut. Breads, for any of you that are fans of carbohydrates, we use fermentation for that process as well, and we will talk about how that works. So here are a lot of other fermentation products shown in this image. If you are a lover of cheese, that is a product of fermentation. Yogurt, even for those of us nail polish right now, rubbing alcohol is something that is very difficult to come by in stores. That is also a product of fermentation. So again, lots of food, lots of drink. Oxygen isn't everywhere. Bacteria in our wounds use anaerobic pathways. So many examples of why this is important. So let's start off with anaerobic respiration. And let's talk about how is this different than aerobic respiration. So the main difference between anaerobic and aerobic respiration is that we use a different final electron acceptor. So in aerobic respiration, what's our final electron acceptor? Hopefully you are yelling at the screen and you are telling me that it is oxygen. So oxygen is our final electron acceptor in aerobic respiration. In anaerobic respiration, we can actually have a more diverse electron acceptor. So we can have SO4 2-, which is sulfate. So that's an example of a pretty commonly used one. We can have NO3-, which is nitrate. There are a multitude of other ones. If you ever take microbiology, we go into some of the other ones that can be used. So microbes tend to use a whole bunch of diverse ones, and that can result in a bunch of different final products that can be formed. But as a whole, we tend to use inorganic substances as a final electron acceptor. And the SO4 2- and NO3- are just two examples of final electron acceptors that can be used. Okay, so what does this mean in terms of cellular respiration? So we have a final electron acceptor. Do you think that that's really going to affect anything else? The short answer is no. The long answer is it has minor effects, but for the purposes of this class and for the purposes of what we're talking about, we can say that everything is the same except for the final electron acceptor. So what that means is we still have 4 stages. Stage 1, still glycolysis. So recall when we first talked about glycolysis, we said that glycolysis is an anaerobic process. It doesn't require oxygen. So in anaerobic respiration, that can happen because we don't need oxygen for that step. We still have formation of acetyl-CoA. And again, we don't need oxygen for that step. For stage 3, we still do the citric acid cycle. And last but not least, we still have the electron transport chain and chemiosmosis. Again, this is the part that is different. Instead of using oxygen as our final electron acceptor, we instead use an inorganic substance. So other than that, everything else is the same. So let's talk about, okay, what kind of organisms use anaerobic respiration? So it tends to be many of our prokaryotic friends. So many bacteria and archaea, or archaea, tomato. And one of my favorite examples is an archaea that is shown here on the right. So here is M. smithii. And M. smithii is an archaea. And I always spell archaea wrong. Maybe one day I'll be able to spell it correctly. It is also something known as a methanogen. And it is found in the human gut and in ruminant guts as well. And so a ruminant is any sort of mammal that gets nutrients from plant-based foods. So think cattle, sheep, deer, giraffes. And so this methanogen is found in our guts and in their guts. And what it does is it helps break food down. And so you might be wondering, why is it called a methanogen? What's up with that? So it turns out the reason that it's called methanogen is because it uses CO2 as a final electron acceptor. And this is, of course, instead of oxygen. And when it uses CO2 as a final electron acceptor, it actually produces methane. And so methane, of course, is one of the gases that is found in farts. So in this picture here, I tried to find the most ridiculous picture of a cow farting. The reason that we have farts and cow farts is partially due to this methanogen, this archaea that is found in our gut that is using CO2 as that final electron acceptor, and then it is producing methane when it uses that CO2. So I thought this was a really fun example to, again, illustrate what kind of organisms are going to be using anaerobic respiration. All right, so that's about all we're going to chat about when it comes to anaerobic respiration. And the reason for that is, again, it's very similar to aerobic respiration. So my challenge to you is to go ahead and draw anaerobic respiration. So just like we drew aerobic respiration and we drew each of the stages, I challenge you to draw all 4 stages of anaerobic respiration, and you can use M. smithii as inspiration. And so rather than our oxygen as a final electron acceptor, you're going to have CO2 and it's going to produce methane. And methane is, it is not CH3, it is CH4. It's helpful if you don't drop one of your hydrogens. So methane is CH4. So again, I challenge you to go ahead and try to draw anaerobic respiration using CO2 as a final electron acceptor. All right, so now we're actually ready to head into part 2 of this lecture, which is all about fermentation. So in fermentation, actually many different kinds of fermentation exist. So you'll notice that the first step is one that we're familiar with, and that is because glycolysis is an anaerobic process. So we start with glycolysis, which means we take glucose and we make pyruvate. From there, this pyruvate, depending on what we do, we can get different types of fermentation. So we can have fermentation that results in wine. We can have fermentation that results in gas gangrene. So again, if you take microbiology, we'll actually talk a little bit about gas gangrene and how that happens. But many microorganisms do this. So the key point and the reason that I'm showing you this image is that many kinds of fermentation exist. And you can actually have an entire course just on fermentation. And Hanover, if you're interested, the chemistry department does actually have an entire course just on fermentation. But today what we're going to talk about is we're going to talk about two types of fermentation. We're going to talk about alcohol fermentation and lactic acid fermentation. And there's going to be a key point that we will see as we go through these two types of fermentation, and that is that fermentation as a whole is very inefficient. So whereas in aerobic and anaerobic respiration, for the most part, we tend to get a lot of ATP formed. It's a pretty robust process and it's pretty efficient. Fermentation, as we will see, not so much. We don't get a lot of energy management molecules. So what we're going to do is we are going to start with alcohol fermentation. So in alcohol fermentation, we have two stages. The first stage is glycolysis, and the second stage is something that we call NAD+ regeneration. Okay, so let's go ahead and draw that here on the right of this page. So we're going to start by drawing our carbons, and just like we have been in all of our other lectures, we're going to represent our carbons as circles or as balls. So here is our glucose. In glycolysis, we take glucose and we split that and we make 2-pyruvate. So here are our 2-pyruvate. In the process of making those 2-pyruvate, we take 2-NADH and they are, or 2-NAD+, and they are reduced and we make 2-NADH. Stick 2 in there. And we also get a net of 2 ATP. So let's go ahead and draw in our 2 ATP. So this is all stage 1. Now at this point in aerobic respiration or anaerobic respiration, we would take that pyruvate, we would convert it to acetyl-CoA, we would go through the citric acid cycle, and then we would go through the electron transport chain and chemiosmosis. But in fermentation, none of that happens. Stage 2 is pretty short. So what we do is we take that 2-pyruvate and we have enzymes that of course help with each of these steps, just like all of our other processes. We actually have 2 CO2 that come off. Those CO2 come from the carbons from pyruvate. So we end up with two carbon molecules, and these 2 carbon molecules are known as acetaldehyde. So we get 2 acetaldehyde. And let me fix this. There we go. Okay, so now we have started with a 6 carbon molecule. We've gone to 2 3 carbon molecules, lost a carbon from each one, and now we have two 2 carbon molecules. And then because we are making alcohol, we now somewhere have to get our ethanol. So we actually take that acetaldehyde, and that acetaldehyde is converted to 2-ethanol. And it turns out for stage 2, So let's go ahead and label stage 2. The reason that it is called NAD plus regeneration is we actually take that NADH from our stage 1, and that gets oxidized back to NAD plus, which means that that NAD plus can be reused in stage 1 again. So we'll answer this question in a little bit, but hopefully what's going through your mind is, well, why the heck are we doing this? Why not just stop at the end of stage 1? And we will answer that question. But first, let's go ahead and for each of our stages, draw our reaction coordinate diagrams. So let's start with stage 1. I'm going to draw all of our X and Y axes first. Can you tell that I'm really struggling with drawing straight lines? But that's all right. You guys will get the point. In my defense, they look straight from where I'm sitting, but I know that they are not. Okay, so in stage 1, we are starting with glucose, and we end with pyruvate. So that's going to be an exergonic reaction. That exergonic reaction is coupled with taking NAD plus and reducing that to NADH, and that is also coupled to another endergonic reaction, taking ADP plus P and making ATP. So in stage 1, exergonic reaction is coupled with an endergonic reaction. And I drew one too many for stage 2. Okay, in stage 2, we are now taking acetaldehyde and making ethanol. Well, that process is actually an endergonic process. And so that energy to power that endergonic process comes from the exergonic process of taking NADH, harnessing the energy that was put into that bond, and oxidizing NADH and getting NAD+. So one other thing that we can make mention here is we can write down what our final electron acceptor is. So in aerobic respiration, our final electron acceptor is oxygen. In anaerobic respiration, it is an inorganic molecule. And in fermentation, if we take a look at stage 2 here, where we have NADH that is losing an electron, so this is being oxidized, which means that acetaldehyde going to ethanol is being reduced. Acetaldehyde is actually our final electron acceptor. So acetaldehyde is our final electron acceptor in alcohol fermentation. So again, I encourage you as we're going along to start comparing and contrasting all of these different types of cellular respiration. And at the end of this topic, we will actually make a table together. So one thing that I want to add, and what I'm going to do actually, let me see if I can make this work. Okay, we're going to do it up here in this little corner. But one thing that is really interesting, and it's related to what we're talking about, but you're not going to be tested on it. It's just a little extra information that I want to give you. And it deals with what happens when you drink alcohol. So when you drink alcohol, you are ingesting ethanol. And one of the enzymes that you have probably heard of before is an enzyme called alcohol dehydrogenase. And alcohol dehydrogenase takes that ethanol and it actually converts it back to acetaldehyde. And then we're going to take that acetaldehyde and I'm going to just write a few steps and enzymes later. Again, if you take biochemistry, you'll probably talk about this entire pathway, but we take that acetaldehyde and a couple of steps later and a couple of enzymes are involved, and we actually make acetyl-CoA. I apologize. I'm learning a new program here and I accidentally hit a button there. Okay, so here is our acetyl-CoA. Well, acetyl-CoA is something that we've all seen before. So what happens with acetyl-CoA? Hopefully you guys are yelling at the screen and you're telling me that that acetyl-CoA can now go into the citric acid cycle. So it's kind of a cool process that when you drink alcohol, you basically kind of go in reverse. You took acetaldehyde and then you reduced it to make ethanol. Now we're taking ethanol, we're oxidizing it to make acetaldehyde, and then that acetaldehyde, using a couple of other enzymes, can be made into acetyl-CoA. And then that acetyl-CoA can go into the citric acid cycle, which means we can break that down further and we can get more energy management molecules. So again, I thought that this was kind of a cool thing to throw into this class to give you a little bit of perspective of what happens when you drink alcohol. All right, so let's continue on with our discussion of fermentation. And let's answer the question of who uses alcohol fermentation and what is it used for and why is it so important. So probably the organism that is most commonly talked about when it comes to alcohol fermentation is yeast, or Saccharomyces cerevisiae. So in our enzyme labs that we've been working on in this class, you all have actually gotten to use Saccharomyces cerevisiae, and we use Saccharomyces cerevisiae, and we use yeast, and it's the basis for many things. So when we make beer, wine, whiskey, vodka, you name it, most of those processes utilize yeast. So just like in lab where we took our dry yeast, we weighed it out and we added sugar, the same thing happens in beer, wine, whiskey, and vodka. So we take yeast and we add some sort of a sugar source. And we can also add other things like barley and whatnot that will give the different flavors to the different types of alcohols. But the basis of alcohol fermentation and how we make these processes is actually the same basis for making our enzyme sugar mixture that we made in our enzyme labs. The other thing at Hearst that we use alcohol fermentation for is for making dough rise. So on the bottom right here's a picture of someone who is making bread, and then you can see on the right over time that dough has risen. And so that CO2 that we made, so let me go back for a second here. So here where we have the CO2 that is produced, I'll go ahead and highlight that. That CO2 is what actually makes the bread rise. And so oftentimes people when baking will say, oh, does that mean if I'm doing alcohol fermentation and my bread is rising because of the yeast, does that mean that I'm going to get drunk off of bread? It turns out no, because when you actually bake the bread, the alcohol gets cooked off. But now, when the next time you're making bread, perhaps during our quarantine, we're all doing more housework. I know it's something that I've been trying to work on is baking and cooking more. But perhaps the next time you bake bread and you see your bread rising, you can think about alcohol fermentation and you'll know where that CO2 is coming from. Okay, so now that we've covered fermentation, let's do a couple of practice problems. So our first practice problem is going back to this idea of why do we take acetaldehyde and why do we then reduce it to ethanol? Why are we not just stopping at the acetaldehyde step? So again, go ahead and pause the video and see if you can brainstorm and come up with some ideas. All right, so welcome back. Hopefully you have brainstormed some ideas as to why we don't stop. And really the reason that we don't stop comes back to this part of our diagram. If we take a look at stage 1, we are reducing NAD+ to make NADH. In stage 2, we're oxidizing that NADH. Now, imagine what would happen if we stopped at acetaldehyde. The cell would only have NADH. So if we only have NADH, we never get back NAD+. So now if we have no NAD+, we can never do stage 1 because we have nothing to reduce. And so the answer really comes back to this idea of we have a limited supply of NAD+. Just like if you are baking cookies and you only have 3 cookie sheets, you are limited by the number of cookie sheets you have as to how many you can put into your oven. And so in the cell, we have a limited supply of NAD+. And it's why ATP and NADH, why these are constantly generated and used. And it's just the cyclical process because we have a limited supply. And so when it is all converted to NADH, glycolysis would stop. And of course, as soon as we converted some of that NADH back to NAD+, we could restart glycolysis. But that's part of the reason why in alcohol fermentation and then in lactic acid fermentation that we'll see here shortly, why we regenerate that NAD+. So one last thing that we are going to do before we move on to lactic acid fermentation is we're going to do a summary of energy management molecules. So in alcohol fermentation, we have an input of glucose and output. So let me actually write this down. So an input of glucose, for an output, we have CO2, we have ethanol, and we have 2 ATP. So in terms of energy management molecules, we get 2 ATP, we get no NADH because the ones that we make, we reuse, and we don't have any FADH2. So again, super inefficient because we only get 2 ATP. All right, so now we are ready for lactic acid fermentation. And we're going to draw what happens just like we did with alcohol fermentation. Luckily for us, it is very similar. It has 2 stages. You can probably guess what the first stage is. The first stage is glycolysis, and the second stage, believe it or not, is actually the same thing. So NAD+ regeneration. And just like we did on the right of this page, we are going to draw our lactic acid fermentation. So we're going to start with glucose. Stage 1 is glycolysis. So in glycolysis, we're going to split that glucose to 2-pyruvate. In the process of splitting that glucose to 2-pyruvate, we take 2 NAD+ and we reduce them to 2 NADH. And we also take 2 ADP, and we make 2 ATP. So this is stage 1. In stage 2, we are going to take that 2-pyruvate that we made, and we're going to convert that into lactate. So we start with 2-pyruvate, we end with 2-lactate, and just like in alcohol fermentation during stage 2, we regenerate that NAD+. So that NADH that was made in stage 1 is going to be oxidized. That allows us then to regenerate that NAD+ that can be reused in another step of glycolysis. So just like we did for our alcohol fermentation, let's go ahead and draw our reaction coordinate diagrams so we can keep track of what is an endergonic and what is an exergonic reaction. Let's draw our axes first. Okay, so in stage 1, same thing that happened in alcohol fermentation. We take glucose and we make 2-pyruvate. That is an exergonic reaction that is coupled to the endergonic reaction of taking NAD+ and reducing that to NADH, and taking ADP plus P and making ATP. So that exergonic reaction is coupled to those endergonic reactions to power that. In stage 2, when we go from pyruvate to lactate, that is actually an endergonic reaction, and that is powered by taking NADH and oxidizing it to NAD+. So again, just like everything else that we have seen in the past couple of topics, everything that is endergonic is coupled to an exergonic reaction. So the last thing that we're going to do is we're going to figure out what our final electron acceptor is. In aerobic respiration, that's oxygen. In anaerobic respiration, it's some sort of an inorganic molecule. In fermentation, our final electron acceptor was acetaldehyde. And in alcohol fermentation, our final electron acceptor was acetaldehyde. And in lactic acid fermentation, can you all figure out and tell me what the final electron acceptor is? So I'll give you a minute or so to think about it. Okay, so hopefully you have determined that if we take a look at our chart that we just drew here on the right, when we take our NADH, this is oxidized, which is a loss of electron, which means its electron is passed to pyruvate because pyruvate is reduced to lactate. So our final electron acceptor in lactic acid fermentation is pyruvate. All right, so let me know in the comments below if you're able to figure that out and if you got it. And if you are lost and confused, then there are many ways you can contact me, and I am happy to help you and help you walk through this. Okay, so we are actually, believe it or not, almost done. The next thing that we're going to talk about is who uses lactic acid fermentation and why is it so important. So for lactic acid fermentation, there's a couple of different examples. So ones that we've already seen on the right here, we have sauerkraut, we have yogurt down here, we have red blood cells, here's our really muscular person. So it turns out for lactic acid fermentation, there's a lot of different organisms that can use it. And that can range from our muscles. So our muscles, when we are exercising really hard, actually run out of oxygen, and so they will do lactic acid fermentation. And it used to actually be thought that when you get like a stitch in your side or your muscles start to hurt, that was from the lactic acid buildup. But in recent studies, scientists have actually shown that that is in fact not the case. But anywho, so when we exercise really hard, our muscle cells will switch from using aerobic respiration, which is really efficient, to an anaerobic pathway, lactic acid fermentation, where we still get some energy management molecules, but just not as many. And cells that do lactic acid fermentation all the time are our red blood cells. So our red blood cells have no mitochondria. If you have no mitochondria as a eukaryotic cell, you can't do aerobic respiration or anaerobic respiration. You have to use some sort of other anaerobic pathway. And it turns out that our red blood cells, the only way that they obtain energy is through lactic acid fermentation. And then last but not least, of course, my favorite organisms, many bacteria. So many bacterial species will actually use lactic acid fermentation. And some really great examples are lactobacillus species. So lactobacillus species are found in our gut, and they're also used to make yogurt. They're also used to make kefir, which is a fermented milk. So it's basically yogurt in a drinkable form. But these lactobacillus species help us break down food, and we also use them to make food for us. So hopefully I've convinced you that lactic acid fermentation is pretty important. So let's do a quick summary. So in terms of what goes in to lactic acid fermentation, we have glucose that comes in. What comes out, we get 2 ATP and we get 2 lactate. Okay, so we can do our summary now of our energy management molecules. We just said that we get 2 ATP, we get 0 NADH, and we get 0 FADH2. So again, those NADH, the reason that they are recycled and are regenerated to make NAD+, is the same reason that they were in alcohol fermentation. If we didn't do that, glycolysis would stop. Okay, so we are going to talk about 3 practice problems, and then we're going to do a big compare-contrast table of all the different types of cellular respiration that we've been talking about. So the first practice problem that we're going to talk about is about athletes. So many of you in class are athletes, just like many students at Hanover are. And so we're going to talk about a swimmer. So on the left, anyone know who this is? Hopefully you all recognize him as Michael Phelps. On the right, we have a biker, and hopefully you recognize the biker as Lance Armstrong. And so this question is asking you to compare Michael Phelps, who is swimming a 50-meter freestyle. So he is swimming a short distance, whereas Armstrong on the right is biking a long distance. So of these 2 athletes, which athlete is going to rely heavily on electron transport and oxidative phosphorylation to produce ATP in muscles during the race, and which one will not? So go ahead and take a couple minutes to work through this problem, and then when you come back, we'll see if your answer matches up with my answer. So if we take a look at the long distance biker, he is going to be the one that relies heavily on the electron transport chain and chemiosmosis. So he is going to be biking for 50 kilometers, which is a really long distance. He's going to require a lot of energy over a long period of time. And so the most efficient way of getting ATP is through aerobic respiration, and it's through the electron transport chain and chemiosmosis. On the left for the swimmer, for Michael Phelps, he's swimming a short distance. He needs quick energy. And the easiest way and the fastest way to get quick energy is to rely on glycolysis, which is, of course, an anaerobic pathway. And so even though it's not efficient, it gives him that quick energy that he needs to swim that 50 meters. So again, hopefully I have convinced you that in terms of athletes that require or rely most heavily on the electron transport chain and oxidative phosphorylation are going to be the ones that are doing longer distance. And so that's going to be our long distance biker, Lance Armstrong, here on the right. So we've got 2 more practice problems. The next practice problem deals with trying to figure out during stage 1 and stage 2 what is oxidized and what is reduced. So I'm going to go ahead and ask you to pause the video and have you work through this problem and then come back and double check your answer and see if it matches up with mine. So the first part is asking us about glycolysis. So during glycolysis, what is being oxidized and what is being reduced? So when we take NAD+ and we add a hydrogen on there, so we are gaining a hydrogen, gaining an electron, that is a reduction. And so that means NAD+ is reduced. If that is reduced, then that means our other partner has to be oxidized. So glucose to pyruvate is an oxidation. On the flip side, during fermentation, so during stage 2, when we form lactate from pyruvate, we are actually, during that step, taking NADH and it is being oxidized. So NADH gets oxidized, which means that pyruvate to lactate must be reduction. So hopefully your answer lines up with mine. All right, we've got one more practice problem. So here we have a newly discovered organism, and here are its characteristics. So it has a final electron acceptor that is acetaldehyde, and then this organism could be made or used to help make wine. So based on this, what kind of respiration is likely occurring? So in terms of what kind of cellular respiration, we can cross off A. We don't have aerobic respiration happening because our final electron acceptor is not oxygen. For B, anaerobic respiration. Again, we don't have an inorganic electron acceptor, so we can cross that off. For D, lactate fermentation or lactic acid fermentation. Well, wine is alcohol fermentation. It's not lactate fermentation. Maybe if I had said that this organism is helping to make a new kind of yogurt and we're still investigating what the final electron acceptor is, then you could make the argument of lactate fermentation. But because our final electron acceptor is not pyruvate and it's not being used to make yogurt or not being used in mussels, we can go ahead and cross that off. And so that leaves us with ethanol or with alcohol fermentation. All right, so last but not least, what we are going to do is we're going to do a big summary. And in this summary, we are going to compare and contrast aerobic respiration, anaerobic respiration, and fermentation. So I'm going to challenge you to go ahead and pause the video and to see what you come up with on your own, and then see if what you have matches what I'm going to add into our table. So I am going to start with aerobic respiration. Aerobic respiration is going to require oxygen. It has 4 stages. Those 4 stages are glycolysis, formation of acetyl-CoA, the citric acid cycle, the electron transport chain, and chemiosmosis. We're going to write down what the final electron acceptor is, and that is oxygen. And it is a pretty efficient process. So we get anywhere from 36 to 38 ATP. For anaerobic respiration, this doesn't need oxygen. It has 4 stages. Those are the same 4 stages as aerobic respiration. So glycolysis, formation of acetyl-CoA, and citric acid cycle, and electron transport chain, and chemiosmosis. The main difference is the final electron acceptor. So it tends to be an inorganic molecule, and great examples of that are NO3-, SO4 2-, and then we also talked about an example where we can have CO2 in the case of methanogens. And so this process is definitely more efficient than fermentation, but depending on the organism, it can be a little less efficient than aerobic respiration. For fermentation, again, there's no oxygen, so this is an anaerobic pathway. And both aerobic respiration and fermentation are both anaerobic pathways. So they're completed in the absence of oxygen. Unlike aerobic and anaerobic respiration, fermentation has 2 stages. So it has glycolysis and the regeneration of NAD+, and there are different electron acceptors. And so we can split this up between alcohol. So alcohol, its final electron acceptor is acetaldehyde. And then for lactic acid fermentation, its final electron acceptor is pyruvate. And fermentation as a whole is very inefficient. So compared to the 36 to 38 ATP that we get in aerobic respiration, we only get 2 ATP in fermentation. So that is the end of topic 16, which is anaerobic respiration and fermentation. Believe it or not, we're actually almost done with metabolism. The very last thing that we have to talk about is photosynthesis. So our next topic will be all about photosynthesis. Once we have completed photosynthesis, we are actually going to do a modeling activity that is going to involve mostly aerobic respiration, but it will tie in some of the other forms of cellular respiration, and it's going to give you practice drawing those energy coordinate diagrams and give you practice of counting carbons and following the carbons, and give you practice of drawing the endergonic and exergonic reactions and which ones are coupled at which stage. So thanks for hanging out with me and sticking with me through another video, and I hope to see you all soon. Have a wonderful rest of your day. Bye, everyone.